Welcome back to The Fight. I'm Pete Byrne, and joining us now is Chang, the Bayer Professor of Engineering and the Director of the Center for Microfluidics and Medical Diagnostics at the University of Notre Dame. Dr. Chang, welcome to The Fight. Thank you for your time today.
Thank you.
I would like to begin by asking you to please talk about the research that you and your team have been working on during this pandemic.
So we're trying to develop a technology that can actually increase the sensitivity of COVID-19 screening tests. So as you may know, the viral load of a sample varies by a millionfold. They can go from a few viruses per mil to a million viruses per mil. And with the current PCR-based tests and also the antigen-based tests, they cannot go down to below 100 virus per mil. So we are trying to come up with technology that will allow them to do that. And if we were successful, what we can do then is do what's called communal screening. So we can screen a lot of people at the same time, much more rapidly, and also with fewer tests. And
certainly that would be very important. Could you tell us, please, how your technology works and ultimately how it would help in this fight against COVID-19?
So it's size-based. So the viruses are about 50 nanometers to about 120 nanometers. That's about 1,000 the size of a strand of hair. So it's tiny. So we would like to strain the virus and let the rest of the proteins and fluids actually go through in the flow-through. But to design such strainers, you have to have pores at precisely size, roughly about 50 nanometers. Too small, and then they get clogged up, and the virus will be trapped in there. And too large, you lose the virus. So we use a special technology. We use energized ions to bombard polymer membranes to etch very regimented, very uniformly sized pores, and we etch them asymmetrically. So instead of straight pores, they actually have a conic geometry. And because of these designs, we can do very high throughput. In less than an hour, we can reduce 1 to 10 mils of sample down to 100 microliters. And by doing that, we increase the sensitivity tremendously.
Okay, so it's obviously very, very precise. How long do you think it'll take for you to realize this technology?
It actually doesn't take long because I've been working on it for some time.
Okay.
Our goal initially was actually to trap exosomes, vesicles in the blood for cancer screening applications. But it turns out the exosomes are actually the same size as a virus, and there are actually fewer viruses than exosomes. So it's actually a simpler task to actually isolate the virus. So we think it takes about 3 months or so for us to test it. And we intend to test the technology first with lentivirus, harmless virus that we can work on in our lab. And then we'll test it at a clear clinical lab on campus that's actually set up by Mike Frender with the South Bend Medical Foundation. And we'll test real COVID-19 samples at that time.
So those are the next steps in the process, and then how ultimately do you validate this in the end, do you think?
Well, if everything's successful, we first have to get clinical approval, so that we require much larger scale testing, and we have to have companies that can then manufacture these kits. So I founded a company that will actually manufacture these membranes once they're approved. So we're hoping that by the end of this year, there could be actually products in the market that one could use. It will be used upstream of all the current technology. All the current tests will be done the same way, but this particular technology, with just less than a half an hour of time that's invested, could make it much more sensitive. And also you can pool. You can actually take many samples. So you can take 100 samples and test all of them at the same time. So by doing that, obviously, you will reduce the number of tests as required. You can reduce the time. So pool sampling is not possible currently, and that's what we're aiming for. We think if we can do that, that will reduce the number of infections when the next pandemic arrives.
Well, it's fascinating stuff for sure, and obviously very important work. So we wish you the best moving forward, Dr. Chang. I appreciate you taking the time from your important work to join us today. Thank you.
Thank you, Peter.
And coming up next, we're going to speak with the CEO of the Cleveland Clinic in London. That's coming up right after this short break.